I want to talk about age-related affordable gene and cell therapies. Now, some people may think that that's an interesting title. The full potential financial conflict of interest list is on this web page on the lower right there. It doesn't fit on slides anymore, but it's there. So I'm going to go through these topics a little more slowly than usual so that I can explain the logic to really understand why we're doing some of the things we're doing, because they're sometimes counterintuitive. So hopefully you'll enjoy this version. And there's a lot of new topics. These are the 4 key topics, which are, we now have a demonstration, just a proof of concept that we can create cells that are resistant to all viruses, not just the ones we know about. Give a discussion of our progress on making enhanced organs, not just organs for transplants, and then multiplex editing. What's the cutting edge of multiplex editing, and how can we apply it to repeats that have been shown to be involved in senescence? And then finally, and the main topic is about aging reversal and gene and cell therapies. So we have these exponentials for reading and writing DNA, and I'm going to make argument that these also apply to therapeutics. Most people think of therapeutics as doing the opposite, of getting more and more expensive. Rather, this is the story of 20 million-fold reduction in price for both reading and writing DNA due to multiplexing. Multiplexing is different from parallelization, miniaturization, and automation. It is mixing of samples that are barcoded typically. So how do the costs work? How do we change that cost structure so that it's available to everyone throughout the world? So for therapeutics, we have a choice of gene therapeutics, cell therapeutics, small molecules, and proteins. And I'm going to draw a line here where I'm going to try to partially justify, this is a highly biased slide, I'll warn you, for why one might be interested in gene and cell therapies at all. One possibility that has intrigued many of us on the gene therapy side is that you can achieve a once-in-a-lifetime or at least a once-in-a-decade type of treatment rather than a daily dose. Daily dose has the advantage you can adjust the dose, has the disadvantage of compliance and so forth. Genes and cells can tend to be a little bit smarter about what they're sensing about the environment, how they're responding to it, where they're migrating, how they're staying localized, and so forth. Also, with genes, there's an obvious mechanism of action. If you know a pathway, you can target a very tiny fraction of it without worrying about the off targets that occur when you have a small molecule that hits, all of the active sites of a multi-gene family in multiple different tissues, where some of those are good and some of those are bad. So that off target is partly what contributes to failure in phase 1, 2, and 3 of clinical trials. One way of reducing cost is by having better outcomes in clinical trials, better mechanism of action knowledge up front. But the other thing that happens, I'll document that in the next slide, is having a large market size. Now I'm specifically talking about age-related diseases by getting at the core commonality of the aging process, not just the symptoms of one at a time for a whole host of maladies that are age-related. In fact, I would argue that almost everything is age-related when you come right down to it, even COVID-19, falling down and not getting up, and so forth. Now, when we think about gene therapies, we might reasonably think about the first ones that have been approved by the FDA, which are millions of dollars per dose, $2.5 million for Zolgensma, which is actually very cost effective for relative to previous treatment or even lack of treatment for spinal muscular atrophy, 2.5 million. But if you look at 5 of the top 5 vaccines, they're all configured as gene therapy. So 3 of them are J&J, and these other 2 were adenovirus, not the AAV of Zolgensma, but adenovirus, and 2 were lipid nanoparticles. But these were classic gene therapy types, a little different in that you're not trying to avoid immune reaction, you're trying to stimulate it. But anyway, it can be as little as $2 a dose. So you get down from $2 million to $2, I think largely because the number of patients involved, the number of people. So a huge fraction of the world's population has now been exposed to gene therapy and at low cost. So this is slightly counterintuitive until you hear it. Now, here's another thing that's, I think, very strange. It's not immediately in an aging model, but I think it is, since we were just talking about viruses as vectors, there's also viruses can be good and bad. And the vaccines for COVID-19 is a perfect example of that. We have now, the problem is almost all of these are done one at a time. A new strain of SARS-CoV-2 comes out, and you might need a new vaccine, or you might need a new drug as HIV becomes more drug resistant. So what if you had a way of dealing with all viruses at once and for all? Even new ones that occur in nature cannot evade this mechanism. And the mechanism that we've explored now for, I don't know, since about 2004, and finally paid off this year in this bioRxiv paper at the top of this list, is changing the genetic code. First, we changed 1 out of 64 triplet codons, and now 3 out of 64, and soon 7 out of 64. But 3 out of 64 was adequate, as you'll see in a moment. Some of this codon remapping is already in a startup company, or a fairly mature company at this point, called Grow Biosciences, which has a number of modified protein chemistry. But anyway, let's talk about 1 out of 64 triplet codons. This was a fairly wimpy one. We didn't have high expectations, but it did eliminate most viruses all at once, not one at a time, not custom made, but just a generic solution. And this was documented in these papers and in 3 different labs, and 2 different generations within my lab. And these are all different kinds of viruses: single-stranded DNA, double-stranded DNA, single-stranded RNA, and so on. Okay, but not all of them. And so then, and then there was an intermediate one that got a few more, and then this is the one that got all of them. It's still in preprint form, where we recoded 2, in addition to stop codon, which was in the first set, we used 2 recoded serine codons, thanks to Jason Chin's group. But we recoded them from null, which is what Jason had done, to leucine encoding, and this overwhelmed the viral mechanism, the remaining viral mechanisms that we hadn't already handled. And this is thanks to Akos, Svena, and Reagan. Now we'd like to put that kind of multi or all virus resistance and a number of other things into organs, not just to address the desperate failure of society to provide enough organs, to having so many people on the waiting list, dying on the waiting list globally. We want to, in addition to that, we want to have enhanced organs, and some of the enhancement is so that we can make less and less need for immune suppression, ideally eliminate the need for immune suppression altogether. And so a lot of these factors here that we've modified, 42 of them now is our record for germline engineering of pigs. They involve 3 really toxic sugars, toxic to humans, not to pigs, clotting factors, matrix compatibility, endogenous retroviruses. We eliminated all of those, which the FDA considered a big risk for immune suppressed patients. Complement, another blood cascade, and other immune functions. Now, this was in these papers, and we have a number of clinics that are there's preclinical primate trials and clinical trials in humans. And Jim Markman, for example, is running the clinical trial at Massachusetts General Hospital. Luhan Yang, which was one of the originators of CRISPR as a technology in humans, was a student and postdoc in my lab and co-founder of these two companies. So what about extending this from just dealing with the need for organ donors to actually an organ enhancement? You don't want the organ to be, the transplanted organ to be causing problems. So you actually want it to be a little bit better than average organ. There are things that you can do with germline editing of pigs that you can't do in humans, but both because the ban on germline, but also because the difficulty of syncing up the donors and recipients. So anyway, with pigs that get more and more humanized, they also become new synthetic biology. I already mentioned resistance to viruses, but we also know in certain cases in animals how to make them resistant to cancer, senescence, cryopreservation, DNA damage, and so forth. And I just want to quickly walk you through this. So here's a 12-centimeter long vertebrate that can drop down to minus 55 degrees for considerable periods of time. And I'm not even sure we really explored the full extremes of temperature. Once you get down to minus 55, it's not that big a step to get to 3 degrees Kelvin, which this polypedillium larvae can handle due to very high trehalose, where it can get 100% survival. And this, you can see here, rehydrating. So not only is resistant to radiation of gamma rays, and to freezing, but also to desiccation. But just cryopreservation would be adequate. But we also, another thing that very few people know about is that you can get 4 or 5 orders of magnitude improvement in radiation resistance, ionizing radiation, with just 4 genetic mutations in this model organism or industrial organism, each of them contributing a significant amount. That I haven't seen a comparable study done in human cells yet, but we're moving in that direction. And then there are these repeat elements. I've illustrated how one can do multiplex editing, where you can do large number of edits at once to get, say, the pig genome engineered. But there's also, that opens up the possibility that you could work on previously daunting, nearly impossible, people thought, elements that have been implicated by some of these articles here. None of these are from my lab, but I think they're respectable publications, where the long interspersed repeats, line elements, are involved in aging-like syndromes, progeroid, involved in senescent cells and inflammaging. The shorter repeats, and there are more of them, so there's tens of thousands of lines, millions of signs. Ribosomal DNA is a highly functional repeat that, nevertheless, its copy number is very sensitive, is a sensitive component of cellular senescence. So anyway, these are 3 categories of repeats, and there's some more here that we now can and are editing. For example, I've already mentioned the endogenous retroviruses that we had to eliminate for the FDA in pigs. And then we moved on to an even more abundant family. Instead of 20 to 60, 20 to 80 endogenous copies, there are 24,000 line elements, or at least active line elements, containing material that could be active, and we've knocked out all of them, which in this starting in this paper published in 2020, but since then we've unpublished. We've gone on to show that they are knocked out by replacing the A-base editor. Let's see that we here an adenosine deaminase, so A to G transitions are made. That's relatively non-toxic. You can make 24,000 edits simultaneously using a guide RNA that's aimed at, let's say, the changing this A to this G just down below it. So there's on the left-hand side, there's an A to G transition. That's relatively benign, but the nicking and cutting versions of this deaminase are lethal, almost immediately lethal to the cell. But after we've made the 24,000 edits and we now switch to that normally lethal nuclease, it's now no longer lethal because all of its sites have been removed. And this is work. That last bit of work is from Chunting Wu and Raphael, but the preliminary work was Corey, Oscar, Khalid, and Verena, published in this EARD paper. Now, as I think this audience is painfully aware, there are some number of pathways, let's say 10, I've added cancer to the usual list, that you probably have to get all right all at once and completely, not just partially, because if you just eliminate one of them, you might add 2 years to the life expectancy. But if you get them all at once, you might get true aging reversal, and who knows how far that could go in terms of life expectancy. I'll show you some life plots in just a moment. So we have to get all of these right, and they interact a lot. So it could be that a small number of epigenetic changes could get all 10 pathways. It could be that you need more than 10 factors in order to get all 10 pathways. We don't know yet, but I think it behooves us to consider multiple molecules at once. I've already explained why we're doing gene and cell therapy. Now I'm going to justify a particular subset, which is the ones that act at this extracellularly. In particular, see some of these factors you see up at the top. We published a few papers now using combinations of 2 and 3 of these factors here, which are indicated by red, blue, and yellow arrows and Venn diagram: fibroblast growth factor 21, alpha klotho, and TGF beta receptor 2. It's not the normal TGF beta receptor; it's a soluble version of it. So this is not natural in that sense. And we've tried every possible combination of these in a number of studies where we're looking at age-related diseases that have very little in common other than they are age-related. And so hopefully we're getting at the core components. This review article, by the way, is aimed specifically at aging, not just signaling pathways in general. And you can see there's a lot of information, but I think our half full cup is good enough to go forward with engineering. And that's what we did. So I'll show you these 3 factors in Noah Davidson's and our PNAS paper in 2019, and a few others that I'll mention in a moment. And here's 3 of many other tests that we've done. These are physiological tests that look at a high-fat diet, obesity, which can be deleterious to the health, either alone or in combination with diabetes, which is the second test here, an insulin tolerance test. And you can see that the sort of the red, orange, and purple ones are doing the best. This is the FGF21 combinations. Here's going from left to right. The control after kidney model for kidney obstruction, and then the K is Clotho, and the F is FGF21, and then the TF is a combination of the TGF beta and the Clotho, and the FGF21. And again, so for all three of these assays, that combination is proving to be valuable, and other studies since then. So of the 4 studies that we've published so far using gene therapies, 3 of them involved adeno-associated virus, which is a very small vector, but it's nevertheless can deliver these combined therapies, either by putting more than one in the same capsid or having multiple capsids. All 3 of those studies, Noah Davidson was a co-author on, and then he left postdoc in the Wyss Institute in our lab to start Rejuvenate Bio, focused initially on dogs, particularly the spaniel, which has a mitral valve disease. We added that to our growing list of now 7 age-related diseases, and it's going up. This is easy to communicate to the FDA. This is what medicine is about. We don't have to explain to them that aging is a disease. Some people think it isn't. We don't need to convince anybody. We're just focusing on these diseases of aging. And if we happen to get all of them, that's a bonus. And that's what we're aiming for. And then one of them was not AAV delivery, it was cytomegalovirus delivery. This is a much larger virus that can handle more genes as we move up into multiple gene therapy. This is, I think, you see we have these 3 factors which act on the outside of the cells, so they're carried by the blood, they're carried in the blood. They're quite soluble and potent. We've also looked at things that I think are less attractive in a certain sense. These are OCT4, SOX2, and KLF4, which have been proven in a variety of experiments to cause severe reversal of aging, all the way, like from 80 years of reversal to embryonic stage, and anything in intermediate as well. But the problem is they require delivery to every cell you want to impact, because these are not carried by the blood. But nevertheless, we're pursuing all of these in either Rejuvenate Bio or BioViva. And I'll show you some BioViva data in just the next slide. So this was, again, the cytomegalovirus. And here we weren't just looking for reversal of aging, we were looking a possible consequence. Now, I mean, it's guaranteed aging reversal is also longevity. And here you can see the short lifetime of the mouse extended by either TERT or FST, follistatin, out many months. And their previous papers that I'm acknowledging here, pioneers in the field that indicated that we would be expecting positive effects from these genes. TERT being, every gene being dosage sensitive, and don't necessarily want it to be constitutive throughout life, because it could cause go too far and escaping senescence and causing cancer. So I've explained some of the logic for why we think we can get and have gotten resistance to all viruses, all naturally occurring viruses in one organism. We're actively trying to get it to work in additional organisms, human. Right now, we're cranking up multiplex editing. I've already showed some multiplex editing in item 3 in humans of 24,000. We don't need nearly that much to engineer the genetic code in humans. I showed various enhancements we can have to organs. You don't want the organs to fail for the same reason that the organs you're replacing failed. So you want them to be resistant to various physiological traumas and infections and so on. And finally, aging reversal. In a certain sense, all of this is aimed at general health and low enough cost that can be equitably distributed to everybody who wants it. So period, full stop. Thank you.